Hi, and welcome to another video in the Human Transcriptome Array Analysis video series. In this video, we'll be going over how to perform QC in the Expression Console software. The Human Transcriptome Array, or HTA array analysis workflow is summarized in this slide. There are basically two software packages that are used to do the analysis. The Expression Console software, which generates the probe set signals and allows you to do the QC of the arrays, and the Transcriptome Analysis Console, or TAC software, which allows you to investigate the differentially expressed genes and or do alternative splice analysis on the arrays. Today we'll be focusing on the Expression Console software. As I mentioned, Expression Console has two functions. Generates the signal estimates, also referred to as probe set summarization, and it is a quality control reporting tool, basically to help you to answer the question of, did my array work? In order to answer that question, you can look at three different things in Expression Console. You can evaluate the array metrics related to the spike-in controls that you've used, either the hybridization controls, the labeling controls, and the ERCC controls. Evaluation of the performance of the controls can either be done by looking at tables of data or using line graphs. You can also evaluate the signal distribution from the arrays and do array-to-array comparisons. This will be reviewed in a live demo of the software, but before we go to that, I wanted to review the thresholds that are associated with the various array metrics in the following slides. The HTA array passing QC metrics for the hybridization controls, or the spike-in controls. So these have been spiked into all of the assays, and the expected behavior is that bio B should have the lowest signal estimate, followed by bio C, followed by bio D, followed by CRE. If this is not the case, then perhaps something has gone wrong and you should further investigate what is going on the array. For the labeling controls, also referred to as the poly A controls, there is again expected performance. Lice is expected to be the lowest signal, followed by Phi, followed by the Thur, followed by DAP. One important note to note in this case is that if you're using both the ERCC controls and the labeling controls, or the poly A controls from Affymetrix, that they share both the DAP, the lice, and the Phi. So that means that if you put both of them together, then the expected concentrations will not behave as expected. Because DAP is expected to be the highest, even if you add more DAP by the ERCC controls, it is still greater than all the rest of them. But Thur and lice have become mixed up, and so that is a problem when you're evaluating both. And this can be seen in the demo data or the sample data that's provided on the Affymetrix.com website. The third metric to look at is the AUC, or area under the curve, comparing the positive versus negative probe sets on the array. And you can see that the threshold for this is at 0.7, and you can see in this example all of the arrays have a AUC that is greater than 0.7. The final set of controls included on the array are the ERCC controls. Just a quick reminder, remember if you use both the Affymetrix labeling controls and the ERCC spike-in controls, that you're going to disrupt the behavior of both, and there might be some anomalies that are observed. So when using the Mix 1 from ERCC, you should pay attention to the ERCC Mix 1 probe sets. In this case, the probe set signals should increase with step number. For example, as shown here, this set of data has had the Mix 1 probe set spiked in. And you can see step 11 is the lowest, followed by step 12, followed by step 13, etc. on up. The Mix 2 probe set signal will not necessarily correlate with step number. If you choose to use the Mix 2 mix from ERCC, then the Mix 1 probe sets signal will not necessarily correlate with step number, but the Mix 2 probe set signals will correlate with step number. Again, pay attention to the Mix 1 probe sets when you use Mix 1, and use Mix 2 probe sets when you've used the Mix 2 spike-ins. So now we'll be moving to Expression Console. So this is the Expression Console. What you see here is the results of the study after two analyses have been run. You can see your cell files at the top, and the set of exon or alternatively spliced chip files here, and the gene level chip files here. For those of you that followed along with video 1, this is exactly where we left off after video 1. And you can see that all of our array files here are marked as being out of bounds. And so let's, in this case, investigate why this is true. Since we have both the gene level and the exon level chip file sets selected, if we choose to open a report or do any investigations at this point, it will be done for both sets of data. Since we only want to look at one set, let's select a particular group. So we can check an individual group. In this case, let's select the alt-splice group set. So now that's selected. So if we go over here to our toolbox, you can see we've already performed our analysis. So the next step is to look at the array metrics, the QC metrics. Let's start by looking at a table of the data. Okay? So what we have here is Expression Console opens a table. It has the arrays located here, the summary of the test, and then all kinds of various metrics associated with the array processing that have gone along. Okay? Individual metrics here that show up with just a number on a background of white or light blue have passed whatever thresholds are set for the array. As we continue to scroll across, you can see there's a large number of metrics that are visible. And we get along here to two sets of metrics that have backgrounds that are orange, which means that they violate the thresholds that we've set up. In this case, we can see that it's both the lice and the phi. And if you remember from the first part of this talk, I mentioned that if you use both the ERCC mix and the labeling controls at the same time, that you end up messing up the expected behavior of the lice and the phi probe sets. And so that's exactly what has happened here. In this sample data, we used both the labeling controls and the ERCC mix 1, and you can see that in fact, yes, these probe sets are not following the expected behavior. So let's continue to scroll across and see if anything else is in orange, or whether that is our only choice, as that is the only cause for the outliers. So okay, since we know what the problem is with these and it was an expected thing, the QC metrics look pretty good on this case. Okay, next, if you choose, you can actually have a tabular report of these that are transposed. In a future video, I will go over how to customize this report to only display the things that you're interested in. In the first part, during the slide presentation, I actually showed some graphs of the way the metrics look. In order to look at the metrics in a graphical form rather than a tabular form, let's click the Report Metrics line graph button. Here we get a pop-up that allows us to select which metrics we would like to see in a line graph. Any of the metrics that are available in the column headers are available for display. So for example, let's scroll down here and we can see the controls, and we have the spike in controls for the BioB, BioC, BioD, and CRE. If I select all of them and click in the little checkbox, and I click OK, now a line graph will be displayed that shows those metrics here. And again, they're color-coded with the legend up here in the right. If you don't care for the graphing program provided by Expression Console, you can choose to right-click on the graph, and you can save the data as text, and then that can be opened in Excel or your favorite graphing program to make whatever graph you would like out of this. You can also save this as a ping or copy to the clipboard for inclusion in a report. It's also possible to edit the scale to further customize the graph. So now we've evaluated our report metrics either in the table or as a graph. The next thing to look at is the signal distribution. So if we click at the signal distribution, we can generate a signal histogram, as shown here. And so when looking at the signal histogram, you want to see whether there's any arrays that have a very different pattern than the rest of the arrays. And you can see in this case that this all looks pretty good. They're very homogeneous and they have basically the similar signal distribution. And none of the treatments have been phase shifted from each other or anything. In addition to evaluating the signal distribution by looking at the signal histogram, it's also possible to use the box plot display. In Expression Console, two different types of box plots are available. You can either choose to look at the box plots on the intensity values, which is before normalization, or on the box plots of the data after normalization. In general, since most of the analysis use RMA, it makes more sense to concentrate on the pre-normalized data or the intensity data in the box plots as opposed to the signal box plots, as they are after quantile normalization, which basically forces everybody to have the same distribution. So let's look at a feature intensity box plot. In order to do so, we have to have some cell files selected. So let's go back to our study window and select the probe cell intensities. Okay, and now we can click the feature intensity box plot button. After a short delay, a box plot based on the feature intensities for each of the individual cell files will be displayed, as is shown here. And you can see that in this case, we don't have any particular outlier samples. They all look pretty good, pretty uniform signal distribution prior to normalization. So this means that we should be okay to go ahead and proceed, and there aren't any outlier arrays here that need to be removed. After evaluating the box plot, the next thing we can look at is array-to-array comparisons. So if you go to the array-array comparisons, one of the things you can look at are the scatter plots. So let's go back here. In order to look at scatter plots, we have to have some signal estimates, again, generated. So let's check a group. In this case, let's check the, go back to the exon level, and we'll just check 2 of the muscle and 2 of the liver samples, and then we'll ask for scatter plots to be displayed. Here are the display of the scatter plots, and you can see that they are red and green color coded. To mouse over to see which samples are being compared, you just put the mouse on it, and here you can see we have liver versus liver. Not surprisingly, we have not much change. Here we have liver versus muscle, liver versus muscle, liver versus muscle, and muscle versus muscle. So this is how you can look for either sample swaps by not having the expected behavior in your scatter plots. It's also possible to look at MVA plots. I'm going to skip this step and go to the Pearson correlation. So you can either do Pearson's or Spearman's correlation to look at the correlation of the signals between the 2 groups. Let's go back here, and for this we'll go ahead and check all of the chip files. And this produces a heat map. Initially it opens up a bit small, so if you just grab it and stretch it open, you can see here we have a heat map, and you can investigate it and see whether all of the signals from the liver samples are more correlated to each other, and then the muscle samples are more correlated to themselves. One thing that I have always set up on my studies, which I forgot to show you guys how to do, is that you can see here we have muscle, which is one of the attributes for the files appended to the cell file name. So if we go back to the Affymetrix study window, you can see that it's possible to have your graphs and tables sorted by a particular metric and also labeled. And so in this case I have the graphs and stuff sorted by, not sorted at all, and then labeled by tissue, which is how we get the liver to show up at the beginning of each of the graphs. What I'm going to talk about next is the PCA plot, or the PCA graph as it's called here, that basically does a 3-dimensional display of the first 3 principal components after principal components analysis based on signal. So let's click this and get the graph to display. I'm going to resize it a little bit to make it a little bit larger, and I'm going to zoom in to make that, now that I made it larger, fill in. So what are we looking for in a principal components analysis? Well, you can see that based on this we have 2 clouds of data, if you will, or the samples are displayed in space, 3-dimensional space, based on the analysis of the signal distributions. Now what you're interested in is what sample attributes are similar. So if we look first at, we'll color these by technician, and we'll see that in fact if you click in here, it's possible to rotate your graph around. You can see that both Sally and Joe are part of this cluster and Sally and Joe are part of this cluster. So that means that we don't have the 2 things that are driving these 2 data apart is not related to the technician in this case. We don't have a confounding factor variable based on Sally or Joe, and we haven't had any obvious sample swap. So that's, we can color by one attribute and we can shape by another attribute. So in this case, let's try and shape by the attribute we care about, which is actually the tissue. So if we look at this, we can see now that the liver is displayed by squares and that the muscle is displayed by circles. So in this case, when you rotate these around, you can see that the liver samples and the muscle samples are separate, and that the 2 clouds that are formed are basically dependent on the sample type, which is what is of interest to us. The things to be concerned about are if you see these 2 clouds and they're actually based on some other attribute that is less informative to you, technician, date of processing, something like that. Or if you have 4 of your muscle samples are over here and 1 of them is over here and one of the muscle cells that's over here is actually over there, that indicates you have a potential sample swap. So by using both the PCA plot and the Spearman's correlation, it's possible to identify the sort of things as sample swaps and get an estimation, sort of a preliminary estimation of how good or how many differentially expressed genes you can expect. The farther apart your clouds are and the tighter they are, I'm referring to this as one cloud and another, the more separate these 2 are from each other, the higher likelihood there is to find differentially expressed genes between the 2 groups. In addition to the chip files that are produced, it's also possible to export a text version of the data. And so if you go to the export results, any of the open tables and graphs can be exported to a PDF to create a report. And you can dump out the signal summarization values either with or without annotation using these two buttons here. So that concludes the demonstration portion of the Expression Console piece. So in summary, Expression Console can help you answer your question of did my array work. The first is to evaluate whether the array metrics worked. Look at the hybridization, labeling, and or ERCC controls, either as tables or in graphs, to see if they're getting the expected behavior. Second, you can evaluate the signal distribution. Are your signals after normalization, do they look good? Or do you have any obvious outliers from a signal perspective? This can be done either using the box plots or the signal histograms. And finally, you can do some array-to-array comparisons. The expectation is that samples that are similar should have more similar signal distributions, which should show up as higher correlations in the Spearman correlation plots, and they should be in the similar three-dimensional space when you're evaluating the principal component analysis. So in conclusion, I'd like to point out some additional resources that are available on Affymetrix.com. There's the Net Effects Analysis Center, which will give you probe set information or information on genes that are covered on the array. In addition, we have other manuals and software, which include the Expression Console manual itself, Transcriptome Analysis Console manual, and other white papers and data sheets. In addition, there are other videos related to either how to do Expression Console, how to set up the analysis, how to adjust the QC metrics, and then some similar demos on how to use the Transcriptome Analysis Console software. Thank you for your attention.